Good morning, good afternoon, everyone. I'm Hayley Hung from Environment and Climate Change Canada. Today, I'm talking together with Crispin Halsall from Lancaster University about long-range environmental transport of chemicals in the context of local sources. Yeah, so this review is part of a assessment of local sources versus long-range transport, which is conducted under the AMAP forum. And then I'd like to acknowledge the co-editors of the entire assessment, including Cindy DeWitt, Roland Kallenborn, Derek Muir, and Katrine Vorkamp. And also, this long-range environmental transport review was contributed by many Arctic scientists, as well as scientists conducting research in Antarctica, to discuss about long-range environmental transport, knowing that there are also local sources in these remote regions. So to give you an overview of the assessment, the long-range environmental transport review is the first chapter of this assessment, and then followed by several chapters which you have already heard from the lead authors about wastewater treatment infrastructure sources, industrial military sources, current regulations, mitigation approaches, as well as long-range environmental transport versus local sources in relation to climate change last time. And at the very end, there will be a synthesis, conclusions, recommendations, and knowledge gaps, policy implication as well. So without further ado, I'd like to move into the objective of this particular long-range environmental transport review that Crispin and I are leading. In this review, we want to provide an overview of, over the years, of what kind of tools and methods scientists have been using to segregate local sources from long-range environmental transport when we observe certain chemicals occurring in remote regions such as the Arctic or Antarctica. And then we'll also talk about some new consideration about assessing long-range environmental transports of chemicals. For instance, how certain chemicals that originally are not sort of considered as persistent, bioaccumulative, or toxic, or according to their own physical chemical properties, they may not be subject to long-range environmental transport, but we eventually saw them in remote regions such as the Arctic and Antarctica. So we'll talk about mechanisms such as chemicals that can attach to particles and can be transported, like piggybacking onto particles and reaching remote regions. And we'll also talk about transformation products. Certain parent compounds may not be subjected to long-range transport, but they're actually degradation products may be subjected to long-range transport and problematic. So one of the first in-depth investigation of local contamination within the Arctic was for polychlorinated biphenyls, PCBs, which was used in electrical equipment at Arctic radar stations established especially and used between 1955 and 1998, forming what we call the distant early warning dual line stations. There were 63 radar stations across Alaska, Canada, and Greenland along the 66 parallel. This figure shows a mass balance for the dual line station at Cambridge Bay in Canada, showing that PCBs deposited in soil around the immediate vicinity of a station, forming a pollution footprint around the station. And McDonald AL in 2000 estimated that this footprint was approximately a 10-kilometer radius around the station. Although local contamination is clearly more important than long-range environmental transport on this particular situation on the local scale, speaking, the station itself can account for about 85% of the source around this particular footprint. However, if we calculate how much atmospheric deposition of PCBs contributes to long-range transport across the entire Arctic, then we can say that actually long-range atmospheric transport of PCBs across the entire Arctic region in that scale, it actually accounts for 99% of what's deposited in the Arctic. So long-range environmental transport for PCB is actually more important than these local sources such as these radar stations. Knowing that there are local sources, such as monitoring stations, such as that one that we are monitoring in Canada as the master station, which is alert in the Canadian high Arctic at 82 degrees north, which is the world's northernmost monitoring station for persistent organic pollutants. This location was selected to be far away from any local sources, for example, Arctic communities or contaminated sites. However, research stations can also obviously be a source of many commercial chemicals, so such as like at these stations, they are full of computers, there are furniture that can contain flame retardants, etc. So at Alert Nunavut, which is one of the world's like longest running air monitoring program for POPs, we do have a historical time trend for persistent organic pollutants. But how is this station actually contributing to the local sources of contaminants in the region? So we've done in collaboration with Tom Hanna's team to do a mapping study using passive air samplers. Like this one shown on the top left here, with polyurethane foam disc inside a flying saucer device. So at the Global Atmospheric Watch Laboratory, which is like the station here, if you can see my cursor, like this is where the long-term air monitoring measurements are being conducted, and there is a military base here, closer to the shoreline here. So when we did the mapping study, we found that PCBs are actually quite evenly distributed throughout the area of the alert station. So the station itself is actually not a source for PCBs. However, for flame retardant PBDEs, it's very obvious that the Global Atmospheric Watch Laboratory, where we are measuring POPs, the concentrations of PBDs are much higher at this location, especially this number 2 location. And then when we looked at the location of this number 2 site, it's actually facing the door of the GAW lab. So when the door of the GAW lab opens, the PBDs from all these computers inside the lab is actually emitting out and then showing higher concentration. But the good thing is that the concentration of PBDs do drop off quite quickly as we go away from the GAW laboratory. So now I'm going to pass to Crispin to talk about Antarctica stations.
Thank you, Hayley. Hi, everyone. I'm Chris Halsall, based at Lancaster University in the UK. I think as part of our writing the chapter, we thought it was a very useful to include effectively case studies from Antarctica. Antarctica, unlike the Arctic, doesn't have a resident long-term population, indigenous peoples. It doesn't have an industry base, unlike the Arctic, and generally marine traffic, ships, etc., are much less than you would get in the Arctic. So it actually serves as a very useful test bed to look at things like local pollution and to try and decouple that from a long-range atmospheric or oceanic signal. And once you start looking at published studies, you do see a cleaner system as such. And like the Arctic, there is less noise. And that's illustrated in the figure here. There's a number of studies where the intention in those studies is to try and look at the long-range transport signal. And for those people not familiar with the Antarctic, much of Antarctic, the Antarctic continent, is at quite a high altitude. In other words, that ice cap, you can end up with scientific stations and sample stations being up to sort of 3,000 meters above sea level, really quite quickly as you move interior from the coastal areas. And you know, some of those studies are looking at the sort of accumulation of contaminants over a longer time period in a snow or ice or fern core. And it's important in those studies that you try and remove or decouple any local contamination that could interfere with your signal. And that's actually quite useful for determining what exactly is the extent of local pollution. And local pollution here in Antarctica is largely associated with the multitude of scientific bases and how they are supplied through ships, etc. So the figure here illustrates some currently used contaminants, so PFAS. And in the upper right there was a study done at Dome C, which is a study site at about 3,000 meters above sea level. And they were very careful to try and identify what was the contamination associated with the local base. And the first thing is that as you move away from that base, the levels of the PFAS actually drop off really quite quickly. So their background study site was positioned, several kilometers away from the actual base. And the PFAS signal there is remarkably similar to a previous study that had been done several 1,000 kilometers away, again at a high altitude site, but looking at PFAS accumulation over a time series, starting from the sort of industrial period of perfluorochemicals right the way through to current day. When it comes to the sort of local contamination, there's been studies done to look at the sort of footprint that might arise from the workings of the scientific base and all the sort of products and materials that are used within the base to look at that footprint. And so down in the lower left there was a study done at the Casey station, an Australian station. And really the levels that they were observing, both through passive air sampling and also sampling indoor dust and that, were akin to what you might find in a typical temperate urban domestic environment. As you looked at some of the waste effluents, the wastewater, the levels are really quite high of PFAS and brominated flame retardants, but they disperse quite quickly in the local marine environment, and there wasn't any evidence of that signal from the base being picked up in biota. So in this case, the Antarctica is actually a very useful comparator to the Arctic, and where we can quite clearly define the influence of local contamination and use that to as a marker so that we can then identify what is the longer range background deposition of chemicals. And here's an example. So this is from the Korean station located in the Western Antarctic Peninsula, actually on St. George's Island. And this study is quite is interesting because they were deploying passive air samplers and they were looking at contamination around the base, but also trying to determine that long-range transport signal. They're looking at PCB, so the figure there is a plot of the various PCB homologs. And they actually found really quite remarkably high levels of a dichlorinated congener, PCB11. And as it turns out, PCB11 is one of those handful of congeners that is considered to be an unintentional PCB. In other words, it wasn't a major component to the old Arachlor mixes. It actually arises through degradation and the creation of dyes that are used in paints, for example. And they picked up a very strong signal, which they attributed to long-range transport around the southern hemisphere, but also possibly through the use of paints and dyes, etc., associated with the base. As we move to the other side of the planet, and we're back up into the Arctic here, we must be aware of chemicals of emerging Arctic concern. So here we've got the organophosphorus esters, so we've got the sort of chlorinated OPEs associated with flame retardants, and then the non-chlorinated OPEs, which are used as plasticizers. But there are a variety of other chemicals, pharmaceuticals including, included in that, but also particle matter associated with things like plastics and microplastics, whereby ocean transport is an important source into the Arctic. So we tend to think of the long-range transport as long-range environmental transport and not just necessarily atmospheric transport. And this study is focusing on the Svalbard Islands and the major scientific base, which is Ny-Ålesund, and in the Kingsfjorden with the big glacier at the end. And Ny-Ålesund itself as a scientific base and a sort of a seasonal population. We can see from this study, again, deploying passive water samplers, that they're picking up a local signal of organophosphorus, flame retardants and plasticizers that are arising from the scientific base. But again, like some of the studies in Antarctica, the signal is really quite localized, and you can pick up the background signal through glacier runoff, etc., and snow sampling once you move away from the station.
So I'm jumping in here to talk about biotic species being a transport factor of contaminants to the Arctic. Provence has noticed in their publication in 2012 that capelin as a fish type may now be replacing Arctic cod as the key foraging fish, connecting planktons to upper trophic levels, including seabirds, fish that eat other fishes, seals, and whales. So especially the transient species of the capelin that came from southerly locations, they were found to have higher concentrations of legacy organochlorines than similar trophic position resident species. Capelins contain higher concentrations of many current use pesticides, including chlorpyrifos, and then also endosulfan and short-chain chlorinated paraffins relative to other fishes in the Arctic. Similarly, supporting the hypothesis that capelin transfer these contaminants originally from regions closer to agricultural, industrial, and urban sources northwards towards the Arctic and entering the Arctic marine food web. In that case, we can consider capelin as contributing to the long-range transport of POPs to the Arctic marine food chain. Now let's talk about some new considerations when we discuss about long-range environmental transports of contaminants. Firstly, the first example, as I'm going to show, is PAHs. PAHs are toxic substances that are unintentionally released to the environment via combustion. So there are also local sources as things are being burned, or say using wood and coal as a source of heating in the north. However, the PAHs are known to be degrading quite quickly in air once they get emitted into the environment. So the question is that, how come they are subjected to long-range transport to the Arctic? Because potentially they would have reacted in the atmosphere and disappeared. So in 2012, Zhao et al. Conducted an experiment and showed that PAHs can be associated with particles and are shielded from oxidation reaction in the atmosphere during long-range transport. That's why this is one evidence showing that potentially PAHs, even that they are very labile in the environment, they are also subject to long-range transport when they are carried by particle-bound particle substances. So another example is about transformation of PAHs. Nitro PAHs is known to be equally or more toxic than any other unsubstituted PAHs, but they have very short atmospheric lifetime. However, modeling studies by Wilson et al. Have predicted that they can spread across the globe as well as moving to the Arctic because of continued secondary formation from pH precursors through OH and NOx reactions during long-range transport. So this same phenomenon is also applied to PFAS precursors. So in 2023, my team was part of a study to look into novel organophosphate esters, which are transformation products of high production organophosphate antioxidant OPAs. The OPAs were is a high production volume chemical applied to plastics, which are used as antioxidants. So while OPAs are non-persistent, they can transform to OPEs in the presence of atmospheric ozone, and OPEs are persistent. So OPE is actually itself a group of new age flame retardants and plasticizers used in products like electronics and furnitures. And then OPEs, like traditionally, we believe that OPEs found in the environment were originated only from, like, say, volatilization or abrasion, as well as leaching out of OPE containing products, which is a direct emission source. However, although both OPAs and OPEs are being produced and used, you can see in this production volume chart here, say this parent OPA is actually produced, like the production volume is 500 times of its transformation products, which is an OPE. But we never can find the parent compound in the Arctic. We only found the transformation product. For instance, in Air at Alert from 1994 to 2018, you can see this dramatic increase in the transformation product OPE in Arctic air, which is likely related to the rapid growth of plastic production, leading to this increasing use of antioxidants over the past 2 decades. So we are only seeing this in Arctic air. And then how about the rest of the world? Actually, although the production volume of the parent antioxidant is 530 times that of the transformation products, the parent is almost never found or never detected in ambient air at all, and rarely in indoor dust. But the transformation product, OPE, is found all over the world, not only in the Arctic and also in Antarctica. So last but not the least, I'd like to give a little summary of our review. We know that there are local sources of contaminants in the Arctic regions, which form contaminant footprints near populated areas or contaminated sites. Like currently, the long-term monitoring stations were selected specifically to be away from these local sources, so we consider them as true reflection of any Arctic background concentrations, and we have to make sure that these stations stay clean so that we can observe long-range environmental transports. In order to do that, we recommend that monitoring station conduct regular site audits. To use low-cost passive sampling techniques in air and water, apply to site audits to make sure to do a mapping study to make sure that the station itself stays as clean as possible and is not affected by potential local sources. So there are some evidence of delayed release of long-range environmental transport contaminants to the environment. For instance, chemicals that were previously used and emitted reaching the Arctic by long-range transport are now becoming secondary sources as the Arctic starts temperature increases in the Arctic, releasing these contaminants from thawing permafrost and melting glaciers. So last but not the least, chemical risk assessment needs to take into account transformation products that may be even more persistent, bioaccumulative and toxic, and are subject to long-range transport than their parent chemicals to protect human and environmental health. So we'd like to acknowledge all the Arctic and Antarctica station scientists that contributed to this review, and also Indigenous people organizations of the Circumpolar Arctic who supported our research and monitoring in the Arctic. Thank you. So now I'm going to pass the presentation time, the forum, to Roxana.
Thank you, Hayley and Crispin, for your wonderful presentation that also really nicely leads into or introduces a lot of the topics I want to talk about. So within this presentation specifically, I would like to focus a bit more in on the biotic transport and really what Hayley was touching on, potentially biota as transport vehicles or concentrators of chemicals of emerging Arctic concern. Particularly, the focus will be on plastics. So very much like what Hayley and Crispin were commenting on, this is obviously work that has a lot of different partners, and I specifically want to acknowledge the Indigenous hunters who have been supporting this work. But I would like to, for a moment, take a step back and kind of start at the beginning, because really this work was, I guess, what first got us interested in this was very much the organophosphate esters. And now Crispin has already introduced them quite nicely. So OPEs are a pretty varied group of chemicals that we sometimes like to roughly class into chlorinated and non-chlorinated OPEs, where the chlorinated ones are mostly used as flame retardants with increasing production and use volumes following the restrictions and phase outs of PBDEs. And a lot of the non-chlorinated OPEs are more used as plasticizers. But there's a lot of different types of applications, but so very heavily used in plastic-related applications. So these structures here just give you a bit of a view of how different these types of chemicals may be. And again, in terms of physical chemical properties, very often what we see is that the chlorinated ones are a bit more on the water-soluble side, whereas most of the non-chlorinated ones are really more these classic pulp-like properties, although considered less persistent in the environment. So when first we started thinking about OPEs, they were actually marketed as a sort of environmentally friendly alternative to BFRs because of that reduced environment persistence, although they are known to be often relatively toxic. So production volumes increased, and then what ended up happening that particularly for the chlorinated OPEs, we started seeing high concentration in the Arctic, particularly also in Arctic water. And so then there was more and more evidence on toxicity of these. So we started as a scientific community to get worried about these. So that just as a little bit of background. But then, so what we then started doing was to actually start measuring on a wider scale, larger scale, if we see these OPEs in the Canadian Arctic specifically, and this was a publication from 2018, all across the Canadian Arctic, plus a little bit in Alaska as well. And so the interesting thing here was that, unsurprisingly, in most of these water samples, we saw in green mostly high concentration of chlorinated OPEs, so these more water-soluble ones. But there were distinct exceptions to these rules, where we would see at certain stations, here highlighted in red, plus one I missed here, where the non-chlorinated OPEs, so these are really non-polar chemicals, turned up in really high concentrations in comparison to the chlorinated OPEs. And so for us, that just from a chemical perspective didn't make a lot of sense, but we did see these measurements repeatedly and consistently. So we were wondering what was going on in these specific stations. And what we then found when talking to some of our collaborators was that all of these stations had one specific thing in common. They were really close to large seabird colonies. So we were wondering whether that could be a connection to these patterns that we were seeing. And specifically, one of the things we were hypothesizing was, because so Jen Provencher, who Hayley already mentioned previously as well, had done some research on seabirds being potential transport vectors and concentrators for microplastic contamination that they were ingesting and then excreting around their colonies. So we were wondering, so if we think about plasticizers and flame retardants being obviously a huge part of a lot of plastic products, could it be that what we were actually measuring in those water samples weren't in fact dissolved OPE concentrations, but essentially particles that we had extracted, and that we were picking up microplastic signals and the OPEs attached to those that had been transported with the seabirds towards the Arctic. So, and then when thinking obviously about, okay, how does long range transport work? And we all are familiar with this type of image here from Vanya and McKay's study. We were thinking, well, you know, as has been discussed with, for example, some UV stabilizers recently, could it be that we actually kind of need a seabird or a bird as a specific additional transport pathway that could lead to long-range transport of specific chemicals or plastics into the Arctic? So investigate this further. We looked at, well, what about OPEs and other types of plastic additives, as well as plastics specifically in these different seabird species? So, and here we analyzed a couple of different birds, but I want to focus on these two species and two specific locations, and I'll tell you why in a second. So specifically on black-legged kittiwakes and northern fulmar. So why these two? Well, the nice thing with these ones is that, first of all, they have very different behavior in terms of their foraging, and we had co-located bird colonies at the same location. So particularly, we had a sampling location for black-legged kittiwakes and fulmars at Prince Leopold Island in the Canadian High Arctic. And then we had additionally the fulmars in the Labrador Sea, so in the more southerly location. And the difference here is that, so the black-legged kittiwakes are actually very local foragers. So they really kind of stay around their colonies, mostly for foraging. So they are a pretty good indicator for local contamination. Whereas the northern fulmars are more of your long-range forager with foraging ranges of like 200 kilometers plus. So, and what we did was basically we looked at a whole range of different tissue types within these birds and a whole range of different contaminants that might be associated with plastics, including plastics themselves, but also different flame retardants, legacy, as well as novel flame retardants, plasticizers, surface treatment agents, UV stabilizers, and even trace metals. So jumping in here, first, so this is the result of plastic particles that we found. And so what we saw here was that, so in, you always see in red here, these are the black-legged kittiwakes, and then in blue, the northern fulmars. Up here, that is Prince Leopold Island. And then down here, you see the Labrador Sea sampling stations. And obviously, one of the big things we were seeing from the start was that the more southerly birds had a lot higher concentrations of plastics in their stomachs, as well as there was actually a significant difference at the Prince Leopold Island. It doesn't look as much like that on this image, but basically what we could see was that of all of the kittiwakes that we analyzed, there was actually just one bird that had any plastic in its stomach, whereas every single northern fulmar had plastic in their stomach, just at a lot lower concentrations or particle counts than in Labrador Sea. So there was a huge difference between the two species, even locally. So the interesting thing was when we then looked at the plastic additives, we actually saw the exact same pattern. So which then, depending on the different type of plastic additives, of course, we saw differences in specific concentrations. But again, very much this different of southerly birds having higher concentrations than northerly birds. An interesting one that I just wanted to point you to here, these are our non-chlorinated and chlorinated OPEs. So there already we did see an indication that these non-chlorinated OPEs were higher in those birds than the chlorinated ones, but we definitely wanted to look further into this. So what caused these differences? So we did quite a bit of different types of statistical analyses, trying to get a bit to the bottom of this. So one thing that we wanted to look at obviously was location, distance to source, foraging behavior or trophic niche, different types of tissue. I am not going to go into that just because in the interest of time. This is all published, by the way. Sex, we did look at that. Could be important. In our case, it wasn't, but that might also be because we just didn't have exactly the same amount of males and females. And then we were interested in what's the connection potentially to ingested plastics. So looking at location, again, very much birds from southerly latitudes had higher concentrations than northern latitudes, and that was obviously consistent also with previous research by, for example, Avery et al., who looked at various birds from always here northern latitudes to southern latitudes in the different oceans, and always saw that basically the more to the south you get, the higher the concentrations. So, but what was really interesting was that we didn't just see differences in absolute concentrations of contaminants, we also saw pretty distinct differences in the patterns of what type of contaminants we are seeing, where in the more southerly birds, we saw a lot more novel contaminants, so contaminants of emerging Arctic concern than legacy pollutants, than at Prince Leopold Island, which is really up in the high Arctic. And again, that might not be very surprising, considering that Labrador Sea is probably closer, more southerly, closer to active sources, which means that it would be likely that we see more new contaminants in comparison to Prince Leopold Island, which might be more impacted by just, trophic biomagnification of more legacy pollutants. So, but where this became really interesting was when looking at the actual birds that were co-located at the same place at Prince Leopold Island. Because so here again, so these are the northern fulmars at Labrador Sea, where we only had the northern fulmars. And again, we see a lot of chemicals of emerging Arctic concern, bit of legacy pops. But now the fulmars in the high Arctic had essentially the exact same pattern, if overall lower concentrations, than their, species brothers in the southerly location. Whereas the kittiwakes, co-located with these fulmars, had virtually no chemicals of emerging Arctic concern detectable in their bodies. So, and that obviously was a very interesting connection or potential connection to what we had seen in terms of the patterns of plastics that were ingested, where the kittiwakes, again, had essentially no plastics in their bodies, whereas these fulmars, even at Prince Leopold Island, had ingested a lot of plastics. And so now here's, of course, the question, are those chemicals of emerging concern and the plastics coming from the same sources where the birds picked those up? Or could those chemicals of emerging Arctic concern actually have been leaching out of the plastics that the birds had ingested, and the plastics actually being a contamination source of those? So this is something that we've been subsequently looking more into. But then coming back sort of to this question of, so are then these seabirds potential, or is the indication that these seabirds are sources of local pollution or transport vehicles? And so again, what we saw here was really that if looking at all of our birds, in every single one, and even across different tissue types, we pretty much saw that these non-chlorinated OPEs were present in higher concentrations in the different birds than the chlorinated OPEs, very much consistent with the patterns that we had seen in the water samples around the seabird colonies. And this actually held true through here. We looked then at some additional species. This is very new data. We haven't published this yet, but are in the process. But where we looked at a bunch of different species at a bunch of different locations. And this pattern, we see this consistently through all of these species, all of these different locations, that always the non-chlorinated OPEs out-concentrate the chlorinated ones, even though in practically any other matrix, consumer products and so on, we usually see the other way around. So as a, I guess, a summary, so these Arctic seabirds, from what we are seeing, seem to indeed be bioconcentrators for plastic contamination in the Arctic. The location of the seabird colonies is a really important factor in how much contaminants of emerging Arctic concerns we have in the birds. But one of the main factors we're really seeing seems to be the foraging behavior, and in at least for the northern fulmars, there is also indication that the ingestion of plastic seems to really be at least a really important co-contaminant problem, if not even a potential source of contamination. And so this means that it really looks like birds can be considered potential long-range transport vehicles for these chemicals of emerging Arctic concern. And yeah, with that, thank you for your attention.